Rob. Good evening. How are you doing?
Good. I was starting to get a little worried though.
So we'll get started pretty soon.
174, 175.
We should have 700 more. Maybe everyone can do me a favor and go on your Slack channels and announce to your friends that we have mandatory colloquium right now. Doesn't. It is incredibly hot today. I hope you're all staying.
We hit 100 here.
Okay.
Quick administrative question.
Yeah.
Given the topic, are we willing to post this in a public forum yet?
Yeah, so this is, we've embargoed all the sensitive IP. So all the stuff that we're talking about today is ready for public release. Okay.
All right. Are we ready to start?
Yeah, let's. Yeah, so just for everyone's good knowledge, colloquium is mandatory. So you have co-researchers who are not here with us. So please Slack your friends. Colloquium every Tuesday. It's mandatory. Mandatory means that thou shalt show up, and if you don't show up, we're gonna have problems. Okay, so we're gonna get started. Today we have two people from my group who will be giving colloquium. We have Harsha Rajkumar and Gavin Lee. Harsha is a rising senior at Vermont Christian, and Gavin is a rising junior at Doherty Valley. Harsha joined my group when he was a freshman, thinking that he was going to work on neurodegenerative diseases or something of that type, and actually started his time in this group smashing plants. And all jokes to this day about the plants that Harsha made a mess of with all over the laboratory. But over the last few years, Harsha has developed an interest in developing scalable synthetic routes for conjugating small molecules to macromolecules. Sharing some of that with his coworker Gavin, who recently joined the group less than a year ago. A very steep learning curve ever since. So this is one of the few times that we have a colloquium being given by a group member who is actually not so junior anymore. It'll be a lot of fun, I'm sure. Just a full disclosure, we're not going to be sharing some of the protected intellectual property that is moving towards patentable technology for obvious reasons. So the contents that you're seeing today is stuff that we are ready to release that's already protected. So, cool. Gavin and Harsha.
All right, sounds good. So hello everyone and welcome to our colloquium this evening. And today Harsha and I will be sharing our project, our research project that we've been working on. And the title will be the scalable chemical synthesis of protease cleavable linker of MAE and a hotoreleasable linker of protofill toxin for antibody-drug conjugates, otherwise known as ADCs. So to start off, we should talk about the issues with traditional chemotherapy. And the main issue with the chemotherapy that we see in modern medicine today is that they're not tumor specific, which means they kill healthy cells along with the cancer cells. And that's why you see a lot of hair loss in chemotherapy patients, and that's due to the inhibition of fast dividing cells from chemotherapy. You'll also see a weakened immune system for chemotherapy, which leads to a greater risk of conditions like sepsis, as well as late and delayed effects, such as long-term organ damage and even risk of cancer remission.
Okay, so the issues that Gavin just outlined in the last slide can be addressed with ADC technology or nanoparticles. So essentially, these fall under a category of research called drug delivery research. And what this aims to do is to try and deliver highly potent cytotoxic drugs to a cancer cell. That way, these drugs don't end up killing nearby healthy tissue and can focus on targeting just cancer cells. And this can even mitigate some of the problems like hair loss that you find in normal chemotherapy.
And it's also really important to talk about, the scale compared between small molecule and the biologics involved in ADCs. And so you can see here a small molecule such as like protofill toxin compared to, biologics, considering including like a monoclonal antibody. And aspirin here, which is a small molecule, compared to monoclonal antibody, is there's a substantial size difference between these two things. And so I think it's really important to kind of differentiate between these biologics and small molecules and how the scale is very different between these two things. And another thing about monoclonal antibodies is their specificity. And the antigen introduction will lead to progenitor plasma cell selection, which then creates antigen-specific plasma cells. And the myeloma hybridization will create monoclonal antibody-producing hybridoma cell lines, which then create monoclonal antibody production. And as we said before, chemotherapy for cancer has side effects, which creates collateral damage, which is a great challenge in drug delivery, which causes a great challenge in drug delivery, which is specificity and selectivity. And so we can also see antibody-drug conjugates in the clinic, and kind of the process, as we explained before, the monoclonal antibody will target the antigen on the cancer cell surface, which will then release the mechanism for the cytotoxic payload, which will be like the drug, such as MAE, which we'll be talking about later. And kind of the ADC approach, what is an ADC? Well, an ADC consists of three important parts, which first includes the antibody, which allows for targeted drug delivery because the antibody will attach to an antigen only found in cancer cells. And then we'll have the linker, which is usually degradable, and it links the antibody and the drug. And then finally, we have the cytotoxic payload, which is a drug and can destroy cancer cells, but again, is very toxic on its own, and as a result, requires an ADC. And as I said before, the mechanism for an ADC is involved in kind of this three-step process in which the antibody will bind to the cell, the antigen found in the cell, the ADC will undergo endocytosis, and then the linker will cleave, releasing the drug. And again, this is a very big simplification, but that's kind of how you see the mechanism working in an ADC. And currently, there are a lot of FDA-approved ADCs, such as gemtuzumab and trastuzumab. And here's a more inclusive list of all of the ADCs that are found that are approved by the FDA. And you can see there's a lot of recent approvals as well in 2021. And so again, today, a lot of challenges that we see in ADC technology also include the bioconjugation strategies. And so a lot of ADCs are made, but we don't really know how much drug or the quantity of the drug that is attached to the antibody. So as a result, the drug to antibody ratios are very unknown, and you can have a very impure mixture when you're synthesizing these antibody drug conjugates. And a second issue is, kind of the release strategies that you can see with ADCs. ADCs can cleave or not cleave at all. And, these cleavable methods include, cleaving with enzymes or cleaving with UV light or even infrared light. And so these things are very great challenges in ADC technology.
Okay, so now we can talk about some of the release mechanisms of prodrugs. Okay, so some release mechanisms of prodrugs include enzymatic, photoreleasable, and some are even glycosylated. So before we go into the different release mechanisms, we need to understand what a prodrug is. So a prodrug is a small molecule that is an inactive form of a drug. And essentially what happens is when it reaches its target in the body, then it'll activate by means of cleavage of a chemical group, and that'll allow it to reach its full biological potency. And so you see that a lot with cancer drugs. For example, the glycosylated prodrug you see at the bottom on the pink bar, that's a drug called glufosinamide, actually a nitrogen mustard, which is incredibly toxic. But because it's a conjugate to a sugar, it helps it reach a cancer cell because cancer cells uptake glucose and sugars far more than regular cells, and it helps it be more specific. So as you can see here, an inactive form of a drug would be a prodrug, and a chemical entity will fall off. And so some of these release conditions can be light, pH, enzymatic, or hydrolysis. And yeah. So our group first started off by trying to understand the mechanism of a commonly used photoreleasable protecting group called the 2-nitrobenzyl ester. And what you can see in the left is a nitrobenzyl ether that you can see. And what that does is when you conjugate it to a chemical group, for example, an alcohol, and you create an ester or an ether, under UV radiation, that bond will break. And so we wanted to harness this technology in creating novel photoreleasable prodrugs. So before we could do that, we needed to really understand the kinetics of this nitrobenzyl group. So we studied different groups such as electron withdrawing and electron donating groups on the benzyl moiety of the nitrobenzyl protecting group. And so you can see 3 of the electron donating groups we studied, as well as the withdrawing groups. And we wanted to see the rates of if these different groups will increase or decrease the rate of photocleavage. Yeah. And so you can see the what we found was the most electron donating would increase the rate of photocleavage, as you can see by the TD-DFT calculations. Yeah. So some future studies we wanted to focus on is to try and study the benzylic protons and how benzylic substitutions can affect the rate of photocleavage as well. So you can see by studying the kinetic isotope effect, which essentially we can replace one of the protons on the benzylic position of the nitrobenzyl group with the deuterium. And if the chemical reaction does, or the photocleavage does slow down, that would tell us that the benzylic proton or the benzylic position does have an impact in the photo release, because deuterium by kinetic isotope effect should actually slow down chemical reactions. And if the reaction does slow down, that confirms that the benzylic protons or benzylic position is important in the photocleavage activity. And so we can assay the photorelease via mass spec or HPLC. We can see if these groups that we prodrug to, and we can notice their masses upon photocleavage. Okay, so we wanted to apply this technology into prodrugging a cancer drug called photofluorotoxin. It's not really used in treating cancer patients because of its toxicity, but we wanted to aim to prodrug this compound with a photolabile group in hopes to increase its pharmaceutic potential. And so what you can see is one of our group members had docked photofluorotoxin's prodrug to a tubulin, which is photofluorotoxin's main mode of action. It stops the proliferation of tubulin in cancer cells. And so we can talk about, and what we found was the prodrugs actually bind better to the tubulin, which is not necessarily a good thing because prodrugs should have should have less biological activity. But that also, it's a win-win situation because now we understand that these can be potential analogs for podophyllitoxin. And so you can see two of the prodrugs that we've created based on our initial studies where we studied the different substitutions on the aromatic system. We found that the dimethoxy groups would increase the photocleavage. And so you can see on the bottom, that prodrug has a dimethoxy group. And yeah, so we're hoping to study how it can, MTT assays on cancer cells to see if the prodrugs have limited activity and upon photoirradiation, it has more activity. Okay, so an overview of our antibody-drug conjugate technology. So initially, we wanted to do something really ambitious and bring in a photocleavable linker to an antibody because this is something that's not really been explored. And if we can release the drugs at will while still having an antibody direct the drug to a cancer cell, that gives ultimate complete specificity to delivering drugs to where it's intended to go to. And it decrease the negative impacts of chemotherapy that we see today.
Okay, so now I'll be talking about kind of our second project, which is also MMAE, and this stands for monomethyl auristatin E. And MMAE is a small molecule potent antimitotic drug, and again, such as similar to podophyllitoxins, also a tubulin inhibitor, and it's also very toxic to the human body. And you can see the structure of MMAE. And so our first MMAE linker that we were working on, also known as a generation 1 MA linker, is a non-cleavable linker, which means that the linker itself does not cleave, but instead kind of relies on like the biological aspects of the cell and relies on antibody degradation in order to release the antibody drug into the cell. And for the chemical synthesis of generation 1 linker, we kind of use Hobbit and PIBOP, as well as room temperature stir and solvents acetonitrile and diisopropyl ethylamine for our reagents. But however, we get 30% yield, which Harsh will explain, why that's kind of bad in the next slide.
Okay, so this is an uncleavable MMA linker, as Gavin just mentioned, but 30% yield isn't really optimal, especially looking at how MMA is incredibly expensive. So when we're working in the lab and we want to create this linker, we only get about 30% yield, which is not optimal, and yet we waste MMA starting material, which is pretty expensive. And so the way this reaction works is we're supposed to create, it's an amidation reaction. You can see how you form an amide bond to the secondary amine and you form a tertiary amine. And so we have to use a coupling agent. For example, there's many, which I'll go into, but we used Hobbit and PIBOP because they're relatively stronger coupling agents. And even then we only got 30% yield. So a study that we wanted to do is testing different coupling agents, which I'll talk about later. Okay, so here you can, this experiment we just did 10 minutes ago before the colloquium, but this is something really interesting. We found out that nucleophilic catalysts, which is really important in creating in linker chemistry. For example, there's many like DMAP or pyridine. These catalysts are important in creating or linking a linker to a drug. But the problem we found is that the greenest nucleophilic catalyst, as well as the most efficient that we found in many other reactions, actually decomposes maleimide, which is really important in creating an ADC because that's the active site that conjugates a small molecule to a large biologic. So we aimed, as you can see in the two NMRs that are overlaid, you can see how the two peaks outlined in the red circle of maleimide actually decreases with the DMAP on, which shows that it decomposes over time. So our plan is to study how different nucleophilic catalysts may or may not decompose maleimide, so we can understand how we can optimize linker chemistry later. So this is a study that we've recently started. We're trying to, remember how I said we only got 30% yield from the MMAE uncleavable? Well, we're trying to optimize that, but instead of trying to use lots of MMAE to study the amidation, we bought N-methylvaline methyl ester, which kind of mimics the secondary amine found on MMAE. And what we're trying to do is we're trying to test different coupling agents, as you can see, with all the alphabets that you can see listed, and different nucleophilic catalysts to see which one will give us the highest yield. And hopefully that can help us in the future when we are able to scale up our MMAE non-cleavable linker. So these are some coupling agents, and our aim is to find which coupling agent would work the best. Currently, you can see PIBOP is what we use. And yeah. Yeah, so these are all coupling agents that you normally use.
Okay, so now I'll also be talking about the second generation MA linker, which is a protease cleavable MA linker, which pretty much an enzyme called protease will cleave the linker instead of compared to our first generation, which is non-cleavable. And kind of the synthetic route for the protease cleavable linker involves us attaching a maleimide using EDC and no DMAP, as we explained earlier, and as well as methylene chloride. And we get 50% yield from this first step, which is not bad, but there obviously could be improvements. And then for the second step, we use pyridine as well as DMF and THF. And this step, we might consider like changing how we approach this step specifically because we use 4-nitrophenylchloroformate, and that gives us some problems, especially with like things like yield and how the reaction may decompose in an RBF, especially when introduced to moisture. So we are considering ways to improve this this step specifically. And then for step 3, which conjugates the MAE onto the linker, the synthetic route here states that we states acetonitrile as well as dispropyl ethylamine. But we previously, according to literature, we've used DMF, but that has also given us some problems because DMF is extremely bad for things like PrEP TLC, and so that's why we're considering using acetonitrile instead. And then finally, for the final step, attaching the antibody, we use a pH 7.4 buffer, a monoclonal antibody, and we just literally let it sit there for 45 minutes, and then the antibody will attach. And here you can kind of see the final product of the protease cleavable linker.
So someone on the chat asked us what the difference between non-cleavable and cleavable was, but you can see here, this is an example of an MMA cleavable, enzyme cleavable linker. And what essentially a cleavable linker means is that when you have a small molecule like MMA conjugated to an antibody, once it's internalized by a cancer cell, you still have to deliver the small molecule drug, right? So it has to come off of the antibody. So that's what an enzyme cleavable linker does. Essentially, you can see a protease enzyme will cleave the amide bond, and you do an elimination, and you'll release carbon dioxide, and you'll actually release the free payload. And that's usually how these ADCs are released the small molecule drug upon enzymatic hydrolysis. Yeah. Yeah, and you can go, yeah. So we also aim to study the bioconjugation strategies. So you have a maleimide group, and it's all a magic mystery after that, right? We have to understand how it conjugates to an antibody as well as at what yields. So yeah, we're going to talk about how we study that. So first, we have to take maleimide with any payload, and we have to incubate it with an antibody or a dummy protein, for example, like lysozyme, and for the small molecule actually conjugated to the antibody, and in which location, so which amino acids. So to do that, we can do a trypsin digest. And what that does is you can use the enzyme trypsin that'll cut right between arginine and lysine groups. And knowing the specific sequence of your antibody, you'll know where to find the small molecule conjugated to the antibody via mass spec. And that'll tell us roughly the drug to antibody ratio, as well as where the small molecule is conjugated. Sorry if my audio is breaking up. And yeah, we can, like I stated before, we can also conjugate something like fluorescein to an antibody. That way we don't have to actually look at it by mass spec. We can see the fluorescence of the antibody itself. So I wanted to go back to the ambitious experiment we had in mind. We wanted to create novel photoreleasable ADC linkers. So this is our first take on that. This is a UV labile ADC linker, and we took inspiration from the initial prodrug strategy that we had from the O-nitrobenzyl group. And so what we did is we created everything up to product 8, you can see, with the free alkyne conjugated to the cytotoxic drug protophyllotoxin. And so all we have to do to conjugate to an antibody now is take an azeto and do a commonly used yeah. Gavin, do you want to take this slide? I think my Wi-Fi completely went off for a second.
Okay, sure. So here is a synthetic route for an IR, Styryl ADC linker. And what this linker does is it's kind of similar to our previous ADC linker, which is toxin and is UV releasable. Instead, this will be infrared releasable, and supposedly this will be very, very novel in the ADC world because we really don't see any ADCs with IR technology yet currently. So we've definitely seen a lot of challenges in synthesis. For example, here, we've had a lot of challenges in kind of getting the synthetic route to work, especially with changing like the carbons over here into an H2N. And then there's more challenges in synthesis. As you can see, we've had a lot of challenges as we're trying to synthesize these ADCs. And you can see here, this step involving HCl doesn't work too well. And so we're trying to find ways that we can improve the synthetic route so that we can actually make the ADC successfully. And again here, another example where we use triethylamine and it's not working too well. But so as you can see here, we have to look in retrospection and look and see how can we improve these things? How can we challenge ourselves to make better synthetic routes so that these routes can actually be achievable with our science. Finally, we would like to acknowledge our advisor, Edward New, as well as our group members, the New Group, and finally ASDRP for giving us a platform to do our research and for allowing us to present our research here as well. So thank you so much, and we will now take questions.
So the attendance form is live. If people have questions, can ask.
Any questions?
We have a question about a protease is an enzyme that breaks down other proteins. So how come proteases are being used for this?
Okay, well, I can take that question. That's a good question. There's many different types of proteases. For example, a protease that cleaves the enzyme cleavable linker we have is called cathepsin. And so how these proteases normally work to break down other proteins is they can break down amide bonds that you find between amino acids. And so our linker itself mimics an amino acid, the two amino acids that these proteases normally cleave with an amide bond. And so the enzyme cleavable linkers that we make can be broken down by these proteases because they have that same bond that the proteases normally cleave and to break down other proteins.
If there's no questions, make sure you fill out the attendance form, and then we'll see you next week at the next rendition of. So fill it out if you haven't done it yet. There's 90 people.
Okay, so just checking. So the attendance form for tonight is just the poll?
That's correct.
Okay, thank you.
You know what he said? He said, here, I don't know what